First of all, I just want to thank you all for including me and inviting me for the talk here. So my name is Aditi Falnikar. I work for Columbia University, and I work with mice and rats to understand brain development and diseases. These are the model systems. Model systems, as you know, are particular species of animals that have been developed over many years to answer specific biological questions. And you can use such model systems to understand developments and diseases of human brain in the laboratory. So today's talk will focus on work from Dr. Richard Valley's lab from past few years on understanding brain development, KIF1A's role in it, and how a potential therapeutic strategy for KAND emerged from this work. So I thought I'll start with some broad statements that all of you here already understand, that KAND has a broad spectrum of phenotypes. It can include spastic paraplegia, which is paralysis of the legs, and brain atrophy, which is loss of brain tissue and shrinkage of the brains. So here, the first row shows MRI images from a KAND patient at 9 months of age and 17 months of age. This patient, so the red arrow is pointing to the cerebellum, which is the structure in back of our skull, responsible for balance and posture. And you can see there is a it's drastically diminished in size. The next two rows shows MRI images from a different KAND patient at 10 months of age, 23 months of age, and 4 years and 7 months of age. And in this patient, not only the cerebellum, but also the cerebral cortex has diminished in size. And therefore, these lateral ventricles, which are fluid-filled cavities, okay, I can't point, which are fluid-filled cavities in our brain, those have expanded. This is called hydrocephalus ex vacuo. So both these patients also had paralysis of the legs, and both these patients had the mutation T99M, which is in the motor domain, which is essentially the anomaly at the position 99 in the amino acid sequence of the protein KIF1A. This position lies in the motor domain, so the mutation is in the motor domain. Motor domain is responsible for walking along the microtubule, as you know, and as you know, most of the KAND-causing mutations are within the motor domain. So that suggests that the variant motor or the mutant motor is probably not going to be able to walk along the microtubules. And this was directly confirmed in one study where they found that patient-specific KIF1A mutations, motor domain mutations, did have either reduced processivities or velocities or were completely inactive in the single molecule assays. So if the first top left panel represents wild-type KIF1A, where the slanting lines represent movement, in the successive panels, the lines have become steeper, suggesting that the movement is diminished. In the lower panel, the lines are vertical, suggesting that movement is completely, the motor has been rendered completely stationary because of the mutations. And this suggests that KIF1A mutations probably cause neurological impairments due to defect in the transport function of KIF1A. This is because while one end of the motor is walking along the microtubule, the other end connects to the cargo. And KIF1A is known to transport a variety of cargos. One of the first cargos that was identified for KIF1A is synaptic vesicle precursors. These are the Golgi-derived organelles, which contain essential proteins for building synapses and regulating release of synaptic vesicles. The second cargo that was identified was in C. elegans, actually. It was dense core vesicles packed with essential neuropeptides such as brain-derived neurotrophic factor and neuropeptide Y. And KIF1A also transports nuclei in cells, in radial glial progenitor cells, which are the progenitor cells in our brain, which give rise to most of the other cells of the brain. And this was shown by Valley Lab here. And my next slides will focus on this. But before that, just to orient you to some of the movies and the brain sections that will be in the successive slides, these will be the cross sections through the developing brain. Here, the radial glial progenitor cells are towards the bottom of the brain section, indicated in light purple color. These radial glial progenitor cells send long fibers that extend from the base of the section where those fluid-filled cavities are, the ventricles, to all the way to the brain surface. And these radial glial progenitor cells give birth to neurons, and neurons migrate along these fibers of these radial glial progenitor cells. So if you look at these cells in the live brain tissue, you have to perform time-lapse movies for this. And you can see this cell going up, this cell going down, and you can see it more clearly in this movie here, where the cell is going up, radial glial progenitor cell body is moving up, and here the cell body is moving down. So, cells are oscillating during development. This is a schematic representing migratory movements of all the different cell types in the brain. The radial glial progenitor cells are in light blue color. They migrate or they oscillate towards the ventricle. Their cell bodies move towards the ventricle, and then they move. Ventricular surface is at the bottom. The black line indicates the ventricular surface. So these cell bodies are oscillating towards and away from the ventricular surface, and then neurons are moving along the fibers of these cells. This oscillatory movement is important for brain development because that maximizes the packing density of dividing cells at the ventricular zone, and it also plays a role in determining which cells will become neurons and which cells will become glia. So you can see that all these different movements of nuclei and movements of neurons is going to require mechanical forces, and those are generated by motor proteins like KIF1A. To study the role of motor proteins in brain development, we use this technique in utero electroporation of rodent embryos, where you perform a C-section on a pregnant female rat or mouse, expose the uterus, which contains developing embryos, inject your DNA of interest. So DNA is encoding fluorescently tagged motor proteins, and then deliver a tiny electric pulse to drive that DNA inside the cells. Then you suture the abdominal cavity and let the rat live and let the embryos develop inside the rat, or you can euthanize both the rats and the embryos in 3-4 days and look at the brains. So brains harvested after this procedure have fluorescence. And if you want to study a function of a protein, you have to take it out of the cell and see what happens. So here, the top panel shows a cell which has KIF1A and it's moving away from the ventricular zone. But the bottom panel shows the cell that is lacking KIF1A and is not able to move away from the ventricular zone. And that is the first function of KIF1A in radial glial progenitor cells. These are on the right are the quantifications for it. The second function of KIF1A is in neurons, in migrating neurons. So the top panel shows a cell which has KIF1A, where a cell initially has multiple processes, but over time, the cell was imaged for 23 hours. And over time, the processes are retracted until a single process emerges from a cell, single thick process, and the cell migrates in the direction of that process. However, when the cell is lacking KIF1A, the extra processes are not really pruned, and so the cell is not able to migrate in any one direction. And as a result, on the middle panel shows the green cells that are stuck. The cells are not able to migrate. Interestingly, not just the cells lacking KIF1A are not able to migrate, but the neighboring cells which have KIF1A, they are also stuck. Those are in red color here. And that tells you that there is some kind of communication going on between the cells that are lacking KIF1A and the neighboring cells that do have KIF1A. And that's cell-to-cell communication that Valley Lab has found is actually via BDNF, brain-derived neurotrophic factor, which is one of the KIF1A cargos. And what was actually found was that if you apply recombinant BDNF, which in other words just means commercially available BDNF, you can actually rescue the development of these neighboring cells in these experimental KIF1A deficient rat brain slices. However, BDNF itself is not able to cross blood-brain barrier if you inject it intraperitoneally or if you administer it orally. However, okay, before that, I'll come to BDNF, but so two functions of KIF1A are in radial glial progenitor cells and in migrating cells. When you express this R18W mutation, which is the motor domain mutation, both the functions of KIF1A are affected. However, when you express a truncation of KIF1A, which this fragment, which we have found to bind to the nuclear envelope, only the radial glial progenitor cells are affected, but neurons are not affected. So using our system, you can classify the mutations based on which physiological functions are affected. And coming back to BDNF, so BDNF, as you know, is important for nervous system development and maturation, as well as function. It's one of the KIF1A cargos. And we saw that BDNF is able to rescue, partially rescue development of neighboring cells which do have KIF1A. However, it is not able to cross blood-brain barrier, but this compound 7,8-dihydroxyflavone is blood-brain barrier permeable upon oral or intraperitoneal injection. So it acts like a mimic of BDNF. And importantly, it is also able to restore the normal development of neighboring cells, which do have KIF1A in our experimental KIF1A deficient rat brain slices. So this is very preliminary data, and Marcus has much more data on leg dragger mouse, but we see that it has a leg dragger mouse has hydrocephalus ex vacuo, similar to KAND patients, and it has paralysis of the legs. And I guess it's worth testing our compound in this mouse model. And also NewSite is testing our compound on cells. So this is one of the therapeutic tools for KAND that emerged from potential therapeutic tool that emerged from this study. So I hope my talk has conveyed that it has allowed us to study KIF1A functions, basic KIF1A roles in brain development. You can categorize mutations based on which functions are affected. And finally, there is a potential therapeutic target that emerged from this study. And I would like to thank my mentor, Dr. Richard Valley, our collaborators, Dr. Wendy Chung and Arne Generic, our funding sources, and all of you for listening to my talk. Thank you.
Wow, that was absolutely beautiful. I had no idea that KIF1A was so important for cortical layering. To that point, in our patient population, do we ever see cortical layering defects like lissencephaly or other structural issues like that?
Yeah, so there are in the literature, there are reports of that. In KAND patients that Dr. Chung has collected data from, you see that less. So if the mutation is very severe, you see these defects of migration. We see that in our rat models when we knock down KAND. In leg dragger mouse, we have tried to do layering markers to see if there are any defects in neuronal migration layering. But so far, in my preliminary study, I have not seen layering defects, but I know Marcus is studying that in more detail. So maybe he can explore in more detail there.
Also, I just love how the oscillatory nature of the RGCs. How much KIF1A do we know? Is KIF1A expressed highly in those cells? And,
yes.
And because it stops, does it inhibit the birth of new neurons? Because I know that the mitotic cycle is regulated by that oscillation.
So fortunately, the birth of new neurons is not inhibited, but a lot of KIF1A is required to transport that nucleus up. We know that if we experimentally deplete KIF1A, the cell cycle continues. However, we have done that in our experimental system where maybe 5% of cells are lacking KIF1A. We don't know what happens when the entire brain is lacking KIF1A because the cell division occurs at the ventricular surface. And one of the reasons why these cells are taken away from the ventricular surface is to maximize the packing density of dividing cells at the ventricular surface. If all the cells are just stuck at the ventricular surface, that is definitely going to have some effect. When all in our experimental system, most of the cells are wild type cells. So those are doing that oscillation. And so the cell cycle of these experimental KIF1A deficient cells continues in our system.
Thank you, Aditi.
Thank you.